Okay, so next up we have our phospholipids. So these are overall going to be pretty similar to our triglycerols that we were just, our triglycerides that we were just talking about. Um, so the triacyl glycerols, as I'll remind you over here, is where we have our glycerol with 3 fatty acids. So a phospholipid is going to be a little bit different, in which one of the alcohols is going on the glycerol is going to form an ester with phosphate, PO4 minus 3, which then forms another ester bond with an amino alcohol. So that's down here. So we have 2 fatty acids, and then, or in the case of glycerophospholipids, we have 2 fatty acids connected to glycerol, and then our last alcohol is connected to a PO4 and then connected to the amino alcohol. So the one that I just described is the glycerophospholipid. We also have something called sphingomyelins, in which the actual base is a little bit different. And those, we're not using glycerol anymore. We're using the sphingomyelin, sphingosine as the core. But then again, it still has this phosphorus and amino alcohol on the bottom here. So let's go ahead and talk about what a glycerophospholipid is. So again, we have a glycerol backbone here. As it says, glycerol backbone. We have 2 ester bonds with fatty acids. And then we have our phosphate and amino alcohol. So how that's going to look, let me check my colors. Yep. How that's going to look is we're going to have our oxygen from our alcohol. Whoa, that was crazy, guys. Okay. Our oxygen from our alcohol, that will connect. I'm sorry, let's go ahead and draw the carbon out. We'll have our carbon from glycerol connected to the oxygen. That's the oxygen of the I'm just messing up my colors all here. Okay, carbon of our glycerol connected to the phosphate group. So the oxygen in the phosphate or the oxygen in the alcohol are all shared in this case. We have our phosphorus with an oxygen, a double bond to an oxygen, and then an oxygen with a minus charge. And then another oxygen. So you see in red, PO4, all of those oxygens there. And then that oxygen is then going to be connected to a carbon, and this is going to be the carbon of the amino alcohol. So those are the 2 different ester bonds that we're talking about. So even though it's not a normal ester that we're talking about, it's a phosphoester bond. So let's use pink. So here you can see if you pretended that that phosphorus was a carbon, that's the ester bond. It's the same next to a carbonyl bond that's bonded to an oxygen. So these, both of these together, actually, let's go like this, is called a phosphodiester bond. Whoa. So phospho, because we have that phosphorus, di, because there's 2 of them, and then ester, because it's what's connecting the carbonyl to, or almost carbonyl, phosphorus double bond to oxygen, to our oxygen next to it. There are 2 different versions of glycerophospholipids. They're lectins and kephalins. So the amino alcohol is all that changes in these. So it's determined by whatever amino alcohol is connected at the end there. So if the amino alcohol is choline, which looks like this, let's do it in blue actually. I'm gonna erase this one to make it a little more clear. So we have our nitrogen, and it has 3 methyl groups on it, and then is connected to the carbon chain. and it is charged at a pH of 7.8. I'm sorry, 7.4. Look at my notes correctly. So at pretty much neutral pH, it is charged, a charged species. So what this would look like is you'd have your glycerol that I've shown here, almost all of it. You have your 2 fatty acids, and then you have your phosphorus, your phosphate group, and then your choline amino alcohol. So as you'll notice, this oxygen here is the same oxygen as the amino alcohol. So the one I underlined just a second ago and this oxygen here, they are the same. Then we have to finish this off. Okay. And then additionally, we have these guys over here. The kephalins have, they have 2 different options of what their amino alcohol could be. Can either be ethanolamine, which looks like this. It's almost the same, except now we have hydrogens instead of methyl groups on the nitrogen, or you can have serine, which is a little bit different. It's technically still, it's still overall an amino alcohol, but it's also an amino acid. But still has an amino alcohol moiety, and that, so an amino alcohol is just where you have an alcohol and an amine on the same thing. So you've seen that in all 3 of these cases. Okay, and then lastly, glycerophospholipids contain both polar and nonpolar regions. So this is particularly important in terms of cell membranes. So these fatty acid chains are the nonpolar tail, is what they're called. And then the phosphate group with the amino alcohol is called the polar head. So you can definitely see that it's polar just based off of the fact that it actually even has charged species in there. So it's got definitely a polar attributes to that portion. So they typically, the nonpolar tail and the polar head don't really like to be near each other. They like to kind of get away from one another. So they actually won't look as much like this, how we drew it up here, but will look more like this, where the tail is going to be opposite as the head. So because of this, they like to interact with different substances. So the nonpolar tail likes to interact with nonpolar things. Polar head likes to interact with polar things. And so that's critical to how our cell membranes work. So if you imagine this red circle as our polar head, and then our little squiggly blue lines as the nonpolar tail, you can put lots of these next to each other and end up getting kind of a layered effect. So the nonpolar tails will all like to be near one another. So they all will form the inner parts of the cell membranes. And then on the outside of the cell, which is aqueous, and on the inside of the cell, which is also aqueous, you'll have your polar heads. So the polar portions go towards the polar aqueous media, and the nonpolar sections kind of blob next to each other and make this bilayer, is what it's called.